Okay, let's go ahead and wrap up the fourth part in the lecture on Mendelian patterns of inheritance. So we've talked about Mendel's basic rules. He established dominant recessive types of examples. Those are what we call monohybrid traits. Then we talked about incomplete dominance. We talked about pleiotropy. We talked about polygenic. All these different examples, codominance, incomplete dominance, etc. All these exceptions to the rules. Again, not a criticism to Mendel. He laid the foundation, but there's just so much we're still learning about genetics. So here's an example that doesn't fit Mendel's rules either. It's what we call X-linked inheritance. And this is a very, very unique type of situation. X-linked inheritance involves problems in which, let me get rid of the bold. Okay. The traits are carried on the X chromosome. Alright, so that is the key. The traits are carried on the X chromosome. So when we look at males and we look at females, chromosome pairs 1 through 22 are the same, what we call the autosomes. You get to that 23rd pair, this is where you get males versus females. So females have 2 X chromosomes, males have an X and a Y. So the way X-linked inheritance, and you're also going to see it called sex-linked, same concept. X-linked or sex-linked, same scenario. They're completely associated with the X chromosome. What you're going to see in X-linked scenarios or traits is that the allele is associated only with the X chromosome. So you could have a big A here on the X chromosome, or you could have a little a on the X chromosome. So the key is, excuse me, males have 1 X chromosome, so they only have 1 allele for a given sex-linked trait. Females have 2 X chromosomes, so they will have 2 alleles for that trait. Okay, so the best way to look at this is put it into example. Color blindness, hemophilia, baldness, etc. So certain genetic disorders are associated with the X chromosome, and these things are referred to as X-linked disorders. They are only associated with the X chromosome. So for a male to be colorblind, X little a, Y. That creates colorblind male. For a female to be colorblind, she would have to have 2 little a's. 1 little a on each of her X chromosomes. The majority of X-linked disorders are recessive disorders. Okay, so it has to have a small allele for it to express. So a female who is, or let's go with the male who can see color, X big A, Y. The female who can see color, X big A, X. But what's that second X carry? It could carry a dominant A, and now you have a heterozygous female, or it could actually carry, get rid of that, it could carry a recessive A, and she still will be able to see color because she's a carrier for color blindness but does not express color blindness. All right, so when we look at family trees, guys, if you're worried about an X-linked trait like color blindness, baldness, hemophilia, etc., you need to look at, so guys, here's the guy, here's the male, the square. You need to look at your mother, but more importantly, go up 1 more generation to your grandfather on this side of your family. If grandfather is bald, and he had the X with the little a, then you know your mom inherited that, because dads will always give their X chromosome to their daughter, and then mom inherited her other X chromosome from your grandmother. So at minimum, mom's a carrier for baldness or color blindness or hemophilia. So guys, what are the odds you picked up that X with the little a, or possibly mom's other X that maybe has the big A? You got a 50-50, guys. If your grandfather's bald, you have a 50% chance you're going to be bald as well. Now girls don't necessarily need to worry about the same ratio because they have 2 X chromosomes. So even if the daughter in this example inherited mom's little a, what's dad got? If dad's X chromosome contains a big A, he passes that to his daughter. She's simply a carrier. She carries a gene but doesn't express. Okay, so there's a handful of X-linked disorders out there that we're learning more about. One of the ones of greater concern is this disorder known as Fragile X. So when you look at Fragile X, a piece of the chromosome, the X chromosome here, is actually kind of almost like it's breaking off. And it's not fully bonded and attached to the rest of the X chromosome. So there's pieces of DNA, information there that is almost being lost. Physically, you look at the individual who has it, you don't see a lot of issues with appearance. The face is a bit elongated, the ears, the nose, etc. So what? That wouldn't be any big issue. The bigger concern is the developmental disabilities, the mental disabilities that often come with Fragile X syndrome. So if that's in the family, what are the odds that you've inherited it? Okay, so an exception to Mendel's rules, X-linked inheritance. There's also a lecture or several Word documents posted with all these problems and how to work through them. Did it in the same way. I did a little screencast, the video. You can watch me draw them out. I would highly, I can't stress this enough, highly encourage you guys, make sure you look at those and figure out, make sure you know how to do genetic problems. So, okay, the other thing to mention, and this is a really exciting aspect of science and genetics and biotechnology. We are learning where genes are located on chromosomes. It's called the loci, the physical location of a gene on a given chromosome. So the examples here are from fruit flies. Normal long antennas located at position 0. If a fruit fly is born with short antennas, we know exactly where the mutation has occurred, position 0. If the legs are short, which is a mutation, that change in the DNA has occurred at position 31. So we're starting to sort this information out. And what is becoming really cool, and we'll get into this with biotechnology, let's say you want the gene for short legs, because that's a good piece of DNA. You get a mutated fruit fly or fruit fly that carries that trait. You go in and you actually cut out that gene from that part of the chromosome, and now you have a string of DNA that carries the information to produce those particular legs. Maybe you splice that into some other insect, some other organism, whatever it is, but this is how we're learning to actually take genes and move them from species to species or within a species. Genetic engineering right here. But we need to know the genetics, and this all starts with a basic family tree, looking at physical expression, which is what we call the phenotype, then identifying the genetic information, what we call the genotype, figuring out where it's at on which chromosome, and then what do we do with that information? Cut it out, take that information out, and put that into something else, and get that other organism to express that particular trait. So the genetic code is like a big book, and we're learning how to pull pieces out from one book and put it into another book. Do we need to rewrite part of the book? Is the book expressing, or is there an error in the book that we need to fix or correct? Lots of opportunities. So start thinking about this. As we go into biotechnology, we're going to continue these ideas, and I want you guys to pick a position. Is this a good thing? Is this a bad thing? Should we continue to support this? Should we stop this? Is it going beyond what humans should be doing? Or do we take it further? Those are all things you guys have a very strong voice in, and you need to make your voice heard. Alright, so genetics, make sure we can do these problems. We'll be looking at them when we get into the exams.